So first of all, thanks to the organizer for having me, and thank you everyone for being here despite the early morning of the rainy weather. So my first slide was made yesterday, inspired by Fred's talk. I'm mentioning this article because I remember reading this about the time that, well, I'm a fluid dynamicist and rheologist, and 6 years ago I decided to switch to biology. And this article was among the philosophical articles I read about, what are the good problems to solve in field of biology. And there's an interesting relationship between this article and this perspective and the discussion around biology for physics to the place we are, because the article is around the book of Edwin Schrödinger, What is Life, which I thought is a good excuse to also mention it, because this was another piece of philosophical work or scientific work that I read around 6 years ago, so that influenced me. So there are many good, as you know, questions there. One of the questions that triggered me is about the decay and survival of biological systems. And what I'm trying to address today is some of our most recent works regarding of what organisms do to not to decay or basically survive fluctuating environments. And I hope it's some examples of biology inspiring new physics. So for that reason, and again, based on the talks given this week, I switched a little bit. So I'm not going to talk much about light production or bioluminescence, but I'm going to have a touch on that. Before I forget, the work is done by this cast of characters in my group. Nico is a first student, PhD student in my team, graduating next year early. Gloria and Karen are biology students, marine biologists, and Cynthia was a theoretical physicist in my group. I want to start with this graph, which is one of my all-time favorite graphs. It's a tree of life. Each of these circles that you see here are many thousands of millions of years. Most of the talks we had this week were around here about humans and fishes, maybe a little bit. Zebrafish was also discussed. So what I'm going to focus our much simpler form of life around this area. And in particular, in my group, we are interested in the interaction of life and light. So this is either in the context of producing light or what life does when they absorb light. And I hope this raised some interesting question regarding mechanics at the same time too. So to just give you a little bit of evolutionary introduction. So this is where life was formed just before 4,000 million years ago. Not long after photosynthesis came and on a molecular basis. And now, after all the evolutions, there are at least 3 big groups of organisms that carry photosynthesis. Light production or bioluminescence came a few million years after, but not long after, if you look at the whole cycle. And now there are also a couple of groups of or family of organisms that can produce light, and that's very, very interesting physics, as you can guess, also in that sense too. So today I'm going to talk about 2 systems, biological systems. First is plants. I'm sure you're all familiar. In fact, plants and mechanics have been studied for decades. Such studies are quite common in the literature of mechanics. So this is a plant growing, and what this person, I got it from YouTube, does is that they put the light on the left, plant grows to the left. They put the light on the right, plant grows to the right. So this is phototropism being studied. As you can say, you can look at it as active filaments and look at the mechanics of this. In fact, the study of such goes back all the way to Darwin time. So Darwin, after he came back from his famous trip, he was criticized for the theory of evolution, and one of the things he tried to do was to say plants also related to animals and other things, naively speaking. So what he's done with his son, he wrote 2 books actually on plant movement. This is what I think is one of the first example of particle tracking. So he put glass plates on top of a plant, and during the day he marked where the tip of the plant goes, and he tracks that and correlated where the sun is. This is actually the plant in front of his house that he has studied. What has been less studied is the intercellular mechanics of plants. So plants carry chloroplasts, which is the topic of my first part of my talk, which are these green discs that sits inside the plant. And to understand why chloroplast is a very particular organelle, it's nice to look at a little bit of the evolution of chloroplast or know about it. Just to briefly say it came from this process called endosymbiosis. There used to be a cell by themselves that were engulfed within another cell. So they still carry some genetic information as an organelle, which is quite rare when you think about the biology of the system. What I didn't know a few years ago, that chloroplasts can actually move as an organelle, and the movement of them is quite particular. Janus particles were discussed in the talk of Elena, I guess, and I can make an analogy between chloroplast movement and Janus particle movement, maybe. So this is a spherical cow. For a biologist in the room, I'm more than happy to go to the detail of biochemical signaling. But the way I look at the chloroplast movement is that they look like discs. So here is another great example of them. So they're literally like, if you're thinking about models that include discs, this is an actual natural disc. And they carry this protein on the surface, which I'm showing with these yellow dots, that in a normal condition, they're distributed on the surface of the chloroplast uniformly. They connect with the help of other proteins to the actin filaments that are very different in plants. They're short actin filaments. So you have to consider that if you want to go deep into the biology of the system. So in a normal condition, what happens is the actin filament polymerization leads to the wiggling of the chloroplast. Okay, and if nothing breaks the symmetry, this particle sits there and just wiggle around. But there are at least two sensory systems on the cells, one on the cell itself, one on the chloroplast, PHOT1 and PHOT2, which can get the photo signals and then do something. One thing that they do is that they redistribute this protein, CHOP it's called, on the surface, and now you break the symmetry in chloroplast. Okay, so it's basically, you can, again, thinking about biology giving you physics question, you can think about this, maybe we are now thinking about it as some sort of phase transition on the surface or some sort of MIPS that protein redistribute and then break the symmetry. Okay, so in reality, it looks like this. This is a chloroplast, the red. What the authors here from the WADA group in Japan do is that they shine intense light here, and yellow is this protein that, as you can see, redistribute in one side, and chloroplasts run away from the intense light. You can actually correlate this with the actin filaments, which are shown here in green, and you can see that chloroplasts run away again from the intense light, and as you see, the concentration of actins are more on the other side. It might raise a question.
Filament is toward the chloroplast?
Yes, I was about to say it might raise a question that why it goes. It's a little bit more complicated than that. So I'm not showing the pictures. So what happens is that there are other proteins that basically lock the chloroplast to the membrane. When the active filaments go there, they monomerize the other side, so they kick it to the other side. So it's not like in a mesoscale they're pulling in principle, but they really are pushing. So we got very much triggered by the chloroplast movement. So we were interested to look at it. And so we got this plant in our team. It's called Elodia densa. If you want to study plants, I cannot suggest this plant more. First of all, it's a weed. So you won't have problem growing them. You will have a problem of them growing too much. Another thing is they only have 2 layers on the tissue. So if you're also interested in the epithelial mechanics, there's no more than, so there's one monolayer on the bottom and another monolayer on top. They do 2 different things physiologically, but it's extremely simple in terms of the structure. And all you need is to basically cut a piece of leaf, put it on a microscope, and chloroplasts are autofluorescent, and you don't need to actually mark anything, basically. So you can immediately use fluorescent microscopy, and this is pretty much one of the first experiments we've done. And what we've seen is that these active discs sitting on a monolayer and are completely packed together on this monolayer. And if you come from a glassy background or physics of dense active matter, you start asking questions, why they do this, and is there mechanical properties here that are interesting. So we start looking at statistics of this. Yes?
You're not going to tell us anything about the fluctuations that you see inside one of these objects. Can you briefly comment on what they are?
So each of these objects are chloroplasts.
When you show the movie, you see fluctuations inside. If you're not going to talk about this in the lecture.
Yeah. So we do talk a little bit about it, but the fluctuations come from this bath of actin filaments around them. So this actin polymerization kicks them a little bit. But if the light is dim, they all sit in this monolayer. So this is almost obvious. So this is your plants in-house early morning. And their fluctuation, basically because they're caged, they just do a little bit of fluctuations. But I will tell about the motion. Yes.
So you said that these move away from the light.
Yeah. So yeah, that's a whole thing. So I'm going to talk about photo damage response. So if the light intensity is too much, they're going to run away from it. This is the light that they love, actually. So that's why they distribute themselves to achieve the largest area. That's actually a very good point to mention, maybe now, because that's when they do the maximum photosynthesis. So if the light is good, they spread. If the light is bad, they run away from it. Yeah, I'm going to show you a video in a minute.
Do they fuse, or the actin prevents the fusion of those guys?
I will show you all the statistics. Okay, so we were interested in what the organism is doing here, basically. So we start doing image processing. You pick your favorite machine learning algorithm, segment everything, and you get all the statistics of this monolayer. Something that we first wanted to check quickly was the area or polydispersity more actually of the system, because to get to the disordered glassy system, as you all know, you should have some sort of polydispersity. Otherwise, you might crystallize, and that's not something that the plant probably wants for the chloroplast on a cell level. So if you do get this standard deviation over mean, so it's a polydispersity factor of the system, is suspiciously very close or just above what you get from hard spheres or soft spheres. I know it's a very different system, but it is close enough to be suspicious that is this an active glassy system or not. So this value being reported by many works, including the work of Peter Solesh, Mike Cates, Wilson Poon, and many other works. So that made us think more, is this actually glassy or not? So we started doing a lot of experiments. This is, again, one of our earliest experiments on a bright field microscope. So I like this because it's as simple as it gets. So you put the lift with any microscope, you can immediately observe this in principle. So this is under dim light, and chloroplasts have formed these monolayers on a cell level. And if you don't do anything, so if the light is off of the microscope, they just sit there happily, they form this monolayer as just mentioned, you optimize the photosynthesis. You get as much as light as possible. But if I turn the light on, this is what happens. So this monolayer is now broken into aggregates that I'm going to comment on it after. So this is uniform light on all these active particles, and they break this monolayer and try to run away altogether and find any shade that they can find. This is a whole different story, which I'm not going to talk about, more than happy to discuss. So you can do image processing of this. This is when the light is dim. They just sit there, and you turn the light on. This is the velocity of the particles. This is the moment we turn the light on. It goes up an order of magnitude in average velocity. These are the distribution of the system. So the question I'm going to address is, what are the mechanical properties here? Is there any interesting things there? So this is the same experiment. It's just background reduced. These are the monolayer of that I just showed you. If you look at such experiments, you can collect, I don't know, many thousands of trajectories. Most of the time, this is what you see. So the particles is pretty boring. Particles sit there, they're caged between their neighbors, and they cannot escape. But if you look carefully, once every 1,000 times, you get a particle, like somewhere here, that jumps, and the rest of the monolayer starts to heal and diffuse back actively, and then they basically, you can think about it, some sort of healing. But where the particle goes is they basically, like a glassy system, the particle escape a cage, and then either go one neighbor or two neighbor and three neighbor. I'm going to show you statistics of that in a moment. So this is how the MST of the system looks like. This is for delayed times. Most of the particles in this regime are subdiffusive. Some of them are diffusive, and a very, very few of them, just here, are super diffusive. These are the ones that escape the cage in this chloroplast model. Okay, maybe better is to look at the Van Hofer distribution. This is a step-size distribution, and these are for different delay time. What shows immediately is this exponential tail that you get in a long time, which again can connect to this dynamical heterogeneity, which was also discussed this week. But other small changes in the center where you don't get you get this sharp form of transitions, which is a result of activity to the system. These are not passive particles, these are not thermal particles. They also have some additional activity on it, and that also shows up here. It's quite a minor change because the system is so dense that the activity of the particle is not really significant, okay, unless they jump out of the cage, which results in this dynamical heterogeneity. We made a model. Our model is as simple as it gets. It's zero-dimensional, so it's a harmonic potential, a particle in a harmonic potential attached to elements that we invented. So if the particle vibrates or oscillates in this harmonic potential actively, so the diffusion is also diffusing, that's another detail. But if the strain goes above a critical strain, which is a fitting parameter in the model, it jumps. How much it jumps is also a fitting parameter, T. Okay, so this is what the model gives you. So you're sitting there and then you jump statistically. Okay. And this is what the experiments give you. These are all the jumpy or out of the cage, escaping the cage particles. So if you look, it's very quantized. They either jump 1 diameter or 2, 3, 4 diameter. So this is, again, it's a chloroplast going out of the cage, sitting in another neighborhood, and sometimes they do it actually multiple times when they jump. And again, you're very much familiar. This is what I got from the work of Ludobertier. And if you do it for any glassy systems, this is molecular dynamics of Leonard-Jones particles, you still see something very similar in that sense. The particle that escapes the cage and leads to this dynamical heterogeneity. You can compare the model with experiments and get this data, something that, again, because we have these fitting parameters, what we learn from the model is that the length scale important for the system is exactly this caging length scale, which is exactly the diameter of the particle in the monolayer. There are some more details. For instance, if you do the math, you find out you can actually take our model very close in some asymptotic limits to what is basically continuum time random walk models of glassy system that escape that. So what I want to say basically that it looks like this is the results of the model MSDE. So you can go from solid to liquid in this monolayer by tuning either this critical cage jump parameter or activity or diffusivity of the systems, and these are shown in this curve. And what we found out from the experiments that the chloroplast dynamics, when they form this monolayer, sits exactly between this solid and liquid system, at the border of the phase transition. And that basically gives us an example of that, or at least suggests this might be an example of some sort of living material at this phase transition border. So it's pretty much ready to not only get all the light as an optimized photosynthesis system, but being very much ready to response when the light intensity goes high. So this is extremely important when the light fluctuates rather quickly. There's a lot more to it. There are many time scales in the systems that if you do some sort of control or filtering analysis of the system, you find out it actually filters all the small fluctuations. So if the sun, a cloud goes in front of the sun, they're not going to respond to that. You really need sun to be there strong enough for the plants to respond to. So it's quite elegant. So to end this part, basically what we find, but the thing we found out is that this monolayer of chloroplast gives you an optimized biological system to absorb as much as light as possible for the optimized photosynthesis. I'm not using the term optimized lightly. There's a lot more in this optimization that I didn't talk about. But we also think that this also has this interesting active glassy system dynamic that makes the chloroplast or the plants ready for the mechanical response. So I wanted to finish this part with maybe 1 or 2 questions. First of all, is the models of, a lot of the system glassy models that we talk about are for large number of particles. here's an example that the system is, first of all, confined, and the number of particles is actually very small. It's about 80 to 120 only. So if you're looking for some systems that the number of particles is small and is confined, and that will surely affect the models significantly. Here's the one that nature gives us. The other one that you can ask is that after you stimulate with light and you form this aggregate, what are the mechanical properties of that aggregate? These are rather difficult questions because you go from 2D to 3D, and this is something we try to find out now with 3D microscopy. These are the disks that I was just talking about, and we're trying to find also these nearest neighbors and think about what are the mechanical properties there.
Do you know the mechanical property of a single disk?
No, we don't. They are a little bit squishy. We can see it in the experiment. When they engage, they kind of squishy, but we don't have access to
Cannot isolate that from the cell.
We can't, yeah, we haven't done it. It's potentially doable to isolate chloroplast. There are many protocols for this. Okay, so to show you, I showed you in the beginning the diversity of life. To show you that what I told you is absolutely anything but universal, I'm going to show you an example of chloroplast dynamic in a different cells that we have in our lab. This is a dinoflagellate. This is a single-cell marine organism, so this lives in ocean. And it looks like this. I will come to the shape. And first time we start thinking about what chloroplast does in these cells, we did this experiment. So it's the very first experiment. So you get one of these single cells, you put it on the microscope. This gray thing, I'm going to show you much better videos in a moment, but gray things are chloroplasts of the cell. And you put a lot of light on them, they do this. They actually take the chloroplast inside. So they also have a mechanism to actually remove or shrink the chloroplast somehow. I should mention these cells are all non-motile. So again, there are many examples of nature like us. When the sun intensity goes high, we just run away from sun and many other organisms. But they cannot swim. So they have to develop or evolve some other form of mechanism. This is a very charismatic cell because they're also responsible for light production in the ocean. The video is playing very slowly, but I'm not sure how many of you have seen ocean bioluminescence before. But what is interesting about the cells, they can also generate light by themselves. We also studied this in detail. I'm going to show you only one slide or two about that, but I'm more than happy to discuss it. So this is the cell. It's called Pyrocystis lunula because of the shape of it that looks like moon. Also, unlike moon, is non-motile. I told you already. It's almost a plant, so theoretically, it's leading order a plant. It has all sorts of cell wall and a machinery on the lipid bilayer that gives them that can result to the light production. Again, I'm happy to discuss the biochemical pathway. So the experiments we did for the light production, and I have a note after about that this is the cell. This is a mechanosensing device that we have where we basically fabricate glass micropipette that are bent, and you hold these two pipettes. Both of these are attached to motorized micropipette manipulators, and you can basically control how much to push the cell or how fast to push the cell. So in a rheology language, a strain and a strain rate on your active system, basically. So if you look carefully to what the cell does here, I'm going to push it gently, and it generates light. The zoomed in version is here. So this is the cell. This is 50 microns from here. So the setup gives you full optical access. And this is the pipette that I'm going to squeeze it. And if you do it, it generates light. And look at also the time scale, which is extremely short. And you can keep doing it. So I'm going to make the video get faster from now on. And as you push it back and forth, it keeps generating light. I like this example because it's also a contradictory example of what we call active matter. If you open most of the papers on active matter, they usually say active matter is when you do chemical reaction to do mechanical work. And this is a complete opposite of that. I do mechanical work to get chemical reaction. So I guess we need some sort of revision on definition of active matter. Okay, back to light production. Light response. This was the work, by the way, when I was doing my postdoc in Cambridge with Ray Goldstein. So now we're interested to see how the cell reacts. This is the video that I already shown you. You can do a lot better. So this is another cell, but same experiments. And what I'm going to do is I'm going to shine light and take it off. Keep basically now poking the cell with light. It's up to genetics, but this one is naturally given to you. You don't have to do any genetics. So if you put light, it goes down, it comes back. You put light, it goes down, you come back. So you can actually control it, put a control loop and keep the chloroplast very deep. And this is a sort of response if you quantify it. So this is the area of chloroplast going down very quickly when you shine light, and it comes back with the relaxation time, goes down, comes back, goes down, comes back. So I call it the biological laws. It's really not. So this is on a macro scale, right? So I'm not showing you any detail, which I'm going to say in the beginning, but you can model it also in a very simple model. So we model it as some sort of chemomechanical coupling systems. It's an active void solid, so it's a viscoelastic solid that we have, and now there's an active term. There's a stress term that depends on light, so this is a linear model, can't get easier than that. What we found, and this is something that we found from the theory about the biology of the system, is that when we try to couple this chemistry to the mechanics of the system, we found out we cannot fit it through the experimental results with one chemical. It turns out you need at least a second chemical to be able to fit through it. And that led us to go and look back at the genetics of the system. And we found out they have two photosensors with completely different timescales. And that corresponds to the C1, C2. So that was interesting, that forced us to look at genetics through mathematical models. And you can use such a model. Of course, there are fitting parameters, and you can fit through, but what you get from the model is the time scale governing the system. Okay, this is nice, but does not really answer the main questions we had in mind. So I showed you a GUI material. Yeah.
First, I mean, do you have an intuition for why you need two timescales? And also, like, if what you need is two timescales, could it just be that you have like a two timescale? I mean, the second timescale is in your mechanical model, right? That you just have elastic timescales and one biochemical one. Would that work, or?
Yeah. So the reason we know it's two timescale is not just this relaxation, mechanical relaxation coming back, is that even in the first part, you see some sort of two. So I haven't shown all the experiments. So you can play with the light intensity and even light wavelength, and then you can prove that it's not just a mechanical relaxation. And if you look at the photosensory of the systems, there are two very completely different proteins that sense the light and react to it. And the conformation timescales are very, very different, and interestingly enough, very close to the timescale we find. So we're pretty sure that it's close to that. So what I showed you is a gooey material that squeezes itself to 40%. So the question is, if you're an incompressible liquid or solid, how can you do that? How can you shrink in a confined system? There's a very thick cell wall here, so they don't expand anything. How can you reduce that volume so much? Any guess? Again? You buckle. But how do you buckle? So let me show you. So we did confocal 3D imaging, and what you can see, the system is not really a continuum system. It's a network of materials attached to each other, and you can see the signature from the holes. And basically, when you shine light, if you zoom in one of these, video is not playing very well, but what happens is that all these holes buckle together. And this is very close what you see in metamaterials with negative Poisson ratios, for instance. So the type of chloroplast that these cells develop are fundamentally different from the plant discs that I showed you in the first part. This is a network of material that can be activated and moved around. Okay, so you can do a lot of these experiments, find out how much chloroplast goes down, and find out how much of this relates to Edward's question marginally. That this area that they reduce is also light intensity dependent, for instance, And what we found out, we did quite a bit of survey about the evolution and ecology of these cells. And we started looking at some sort of satellite measurements of the light intensity. And this is exactly what these cells experience. So this is a physiologically relevant zone of light intensity. And the response of the cell is exactly in that regime, what we do in the lab. After that, it's fully saturated. Okay, to just end the talk with a couple of notes about this buckling system. So this is the image of the cell, the chloroplast. You can see the network here again. If you shine light, what happens that these holes close up in principle. This is a better video than the one that I last showed. And this results in the reduction of 40%. And this raises a lot of interesting questions about the topology of the network and also the geometry of the network, which we looked so far to some extent, but very carefully. The way we do this, I should say, the biochemistry of this is also known to some extent, and I'm happy to discuss it. In my team, we call it Taylor-Swift mechanism, after Swift and Taylor, who studied this in 1967. The way we study the topology, we get one of these three-dimensional networks. We fit a network through it. Then we mark the node of the network. And then we let the network evolve over time. And now I have a statistics of these highly connected networks that are activated by light. Okay, and from this you can get a lot of information. For the sake of time, let me focus on just one or two important ones. So what we found out is that if you look at the MST of these nodes of the network, most of them are actually diffusive, with the exception of the few. And if you track where those few are, they're always at the tip, which are also correlated with the length of the network. So all the cell does is basically mainly make the nodes at the top ballistic, and the rest is mechanics. So they can be only that, they can actually shrink it very quickly to the center. So that's also a nice mechanics. Another thing that we found out is that if you look at this as a topological surface, and genus is the topological genus, our reviewer was confused about the term genus because she was an evolutionary biologist. But what happens is that that in the beginning you have a topological surface with genus 100 that goes down as you put light, but it comes back. The shape of the network is different when it comes back, but the topology property, the topological invariant of the system remains the same. And every time we do the experiment, we see this. So this tells us, and it's mainly a question rather than a conclusion, that maybe there is some sort of optimized topology for such a confined network to achieve for such a fast and other response. Okay, let me focus. I mean, we've done also local experiments. So you just shine light in the corner. It turns out the response is actually local, unless you hit the nucleus, which is the most important part, and then the response is actually global. So it's more complicated than even what I showed. All right, so to end this and last part is that what we think is that there's this chemomechanical coupling on a single cell level, but on a network, which resembles some sort of active biological metamaterial. This raises more questions than answers. We still don't know what are the actual mechanical properties of the system. I think we need to develop experimental technique. This was discussed before. I'm not fully convinced we have good, very complete models of active networks yet. So that could be, and again, this is another example, maybe convincing to go to that direction, and this question of optimized topology. So with that, let me end. These are the references, and I'm more than happy to answer any questions. Thank you.